going to be looking at higher fungi, subtitle Ascomycete. So what is it about? So what is this about? So firstly, asco means sac. So Ascomycete have got a sac-like structure called ascus, where mostly sexual reproduction occurs. So as you can see, even the structure resembles that of a sac. So they have a sac-like structure. So the other characteristic is that they are mostly decomposers. So they undergo both sexual and asexual reproduction. Let me reduce the font. They undergo both sexual and asexual reproduction. So we are going to dive deeper into these two, but before that, so they produce ascus. So that is under asexual reproduction. They produce ascus. Which produce spores. So now these spores, specifically for these spores, they are conidia. So conidia. So the name is conidia, like that. So the name of the spores is conidia. That is under asexual reproduction. So asexual reproduction, before we look at the diagram, asexual reproduction here is mostly through fragmentation. So space, but this is not the one that we are going to be looking at. So we are going to be looking at the one that produces conidia. So let's look at the cycle. So let's say we have the ascospore here. So we have, or should I say, not the ascospore. We have the Ascomycete here, so it will be able to produce spores, which we are going to call conidia. So these conidia are released via dispersal, which is a method of transport, or rather means. So it can drop, so these spores can drop, and these spores are called conidia. So when these spores germinate, or rather they grow, they become mycelium. But before mycelium, they have to become what we are going to call hyphae. So hyphae, when they now fuse together, they will be able to form the mycelium, like that. So from there, they will be able to now make the whole Ascomycete. So that's that. So this type of this type is what we call conidiospore, which produces conidia. So the conidia, so just a recap, we have dispersal, so there will be the release of conidia, like that. Then the conidia will be able to grow through germination into mycelium. But first we have hyphae. Hyphae is one strand. When they come as a group, then they'll be able to form what we call a mycelium. So as you can see, so this mycelia contains a lot of hyphae bonded together. Then from there, they will be able to grow and become the ascospore itself. So that is it for asexual reproduction. Now sexual reproduction, on the other hand, is quite involving. So we are going to begin at the ascospores. So the ascospores, or should I say the ascus. So the ascus, the sac-like structure, will be able to produce what we call ascospores. So in other books, they will just say spores. So they will be able to release what we call the spores. then the spores will begin to germinate. So same with sexual reproduction, to become hyphae. So germination is growth. Now these two hyphae are the same. Therefore, the next stage after germination is that they will be able to undergo what we call differentiation. So under differentiation, they will be able to become specialized into the male and the female. For the female, we call it Ascogonia. For the male, we call it Atheridium. Then they will be able to fuse and undergo the process of plasmogamy. So plasmogamy is just the fusion of the cytoplasm. So the fusion of the cytoplasm is what we call plasmogamy. Then from there, so just as we were saying, after plasmogamy, which is the fusion of the cytoplasm, they'll be able to become, or rather develop into this complex structure called ascocarp. So just to write it properly, ascocarp. So this ascocarp, if you extract it, we have this structure, which is the sac-like structure, which we are calling the ascus. So the ascus will now be able to, so remember, this ascus has dikaryotic nucleus, meaning to say it has got two separate nuclei. Then after that, there will be the process of karyogamy, which is the fusion of the nucleus. That's how come you have this nucleus. I've painted it purple and yellow. They are fused together. So from there, they are able to produce the ascospores. These ascospores will now begin again from ascospores. So these ascospores, I've just written this, but others will call them spores. So depending on your lecturer's preferences. So these might be called spores, but this one is more accurate, ascospores. So these ascospores will be released by the ascus. So this is still the ascus. By the ascus, when it is released like that, it begins to germinate, or rather to grow. When it grows, it becomes hyphae. Hyphae will be able to undergo, so this process is differentiation. So differentiation, same hyphae, it has now become specialized into becoming the male and female sex parts. So we have the ascogonium, which is the female, and atheridium, which is for the male. After that, there is plasmogamy, which is the fusion of cytoplasm. So after the fusion of the cytoplasm, there will be the ascus. There will be the formation of the ascoscope. Then the ascoscope, we have a segment or a section, a sac-like structure, which we are calling the ascus. So since they fused together, they formed a dikaryotic cell in which we have two cells present. So now, just a recap, this type of cell formed after plasmogamy is what we call an heterocytic cell, which means it has two separate nuclei like that. So we are here, the ascus will be able to undergo karyogamy, in which there will be the fusion of the nucleus. After that, we are going to have our meiosis. After meiosis, we are going to produce our spores like that. So meiosis is very important here. So after that, our meiosis will take place, in which there will be the division of the chromosomes to produce our ascospores, or rather the spores. So this is basically the ascus. Then the ascus will be able to release the spores, which we are going to be calling the ascospores, and the cycle continues like that. So that's that on higher fungi ascoscopes. Thank you very much for watching. Hope you would like, subscribe, and share for more. I hope this video was helpful. So this is the whole structure. We have conidia. This is, if you see conidia, it has to do with asexual reproduction. So we can have the conidiophore, this one producing conidia. So the spore, which is dispersed like that by dropping or other means, this is able to grow. This is under asexual, germinates to form mycelium, but before that hyphae, which develop into mycelium, which can later be able to regrow into the ascomycete in that case. So this other one, we can begin from here, ascospores. From ascospores we have mycelium. Mycelium becomes specialized to positive, so to positive and negative, but for this one we use these terms. We are going to be using ascogonium, the ascogonium, and the atheridium. Atheridium, which is the male, then the ascogonium is the female. So those are the terms you are going to use for Ascomycete. Positive and negative, you use it for, you're going to be using it for Basidiomycete. So when these fuse, they are able to develop the ascocarp. Ascocarp, a segment of the ascocarp is called the ascus, so which is a sac-like structure. So it will contain dikaryotic, meaning to say it has got two nuclei, two separate nuclei. Even the dikaryotic hyphae, it has got different nuclei like that. Then we are going to have the karyogamy, in which the nuclei will be able to fuse to form one like that. Then the process continues. We are going to have meiosis, which leads to the division of four haploids and all that. So that's basically it for this video. I hope it was helpful, and see you in the next tutorial.